Okay, yeah. So before we get into the hands-on part of the workshop, as I alluded to, we're going to be talking about dealing with a bunch of data that has been collected and figuring out what is going on in a recorded population of neurons. So this is kind of inspired by the stuff that I did while I was in David Anderson's lab, where we have mice running around interacting with each other in a home cage, and we're recording their behavior and their neural activity, and then trying to say something about the structure of both of those things and how they relate to each other. So I will quickly try to go through a couple approaches for interpreting and making sense of neural population data. It's really kind of an exploratory process, and every brain area is going to be different. But these are a couple things you can try, some based on sort of supervised approaches where you have like behavior and neural activity, or stimulus and neural activity you're trying to relate to each other, others that are more unsupervised. So we'll start with looking at the responses of individual neurons and tuning analysis and understanding what cells respond to. And the classical way of doing this is an event-triggered average. So you take, this is the behavior, oops, mouse, chat. Oh, yeah. You take the behavior of the animal and you identify some action the animal is performing or some stimulus that's being presented. And in this raster here, each row is, in this case, a bout of mounting during male-female interactions of mice. And we're aligning this time series data to the onset of mounting. So there were 47 bouts of mounting in this animal. And on the other side, you can align the neural activity the same way, and you can identify neurons that show a modulation of their response triggered on the onset of a behavior. So this particular neuron has an event-triggered average that shows an increase in cell activation triggered on the onset of mounting. In comparison, this light green line here is the same event-triggered average triggered on the onset of sniffing. So this is a quick and dirty way to look for cells that are modulated by something. You can do a significance test, compare the activity before versus after the behavior onset, and see if there's a significant increase or decrease in delta F over F, or firing rate. The one caveat to think about here is that you might see an increase in activity, but it might not necessarily be for the reason you think it is. Just for example, there's an increase in activity triggered on the onset of sniffing here. That could be that this is a cell that is also modulated by sniffing. It could also mean that it's a cell that's modulated by the distance between the animals. And when the mice are close together, even if they're not sniffing, you would see a similar increase in the activity. So event-triggered averages are great, but you need to be aware, especially if you're studying naturalistic behavior, of the fact that behaviors are correlated with each other. They're not independent things. And what you think you're analyzing, what you think is explaining the neural activity, might not actually be the signal that can predict it the best. Nonetheless, the thing you can do, and if you do this for a single cell, you can get an average modulation of that cell by the behavior. You can also do this across a population and cluster cells and identify different groups of neurons that are differentially modulated by a given behavior. So as an example here, I'm showing 3 groups of cells. So here each row is a neuron and each column is a time point, and we're again triggering on the onset of mounting. And we just used k-means clustering, where we took each column, each time point, as an independent variable and each neuron as an observation of that variable. And we clustered the cells based on their response profiles. And you have to tell k-means clustering how many clusters to look for. There are some strategies for picking k, the number of clusters, based on what value gives you the best fit to your data. And once you've found your clusters, you can plot the average across units in a cluster, and you can identify kind of different signals that are present in the neural population. So in this case, we found some cells that were activated by mounting, some cells that were inhibited by mounting, and also some cells that just responded at the onset of behavior. So it's a way of kind of exploring further how signals are represented in the neural population. A second kind of cell-by-cell approach to making sense of neural activity is what's called an area under receiver operator characteristic curve, or an AUROC. This is also called choice probability. And this is, in some contexts, AUROC is more common. And this is a way of, it's basically training a decoder on the activity of an individual cell and saying, if I just had this cell, how well could I distinguish two conditions? So here I'm showing you a neuron that was imaged in VMHVL, an attack-associated nucleus, and we're seeing superimposed over the animal's behavior the trace of the neuron activity, and we have bouts of attack and bouts of sniffing, and this looks like a cell that's a little more active during attack than investigation. So to put a number on this difference, what we can do is we can take these two conditions, all of the attack frames in the data set and all of the sniff frames in the data set, and just make a pair of histograms of the activation of the cell, the delta F over F, on all of the attack frames in red and on all of the sniffing frames in orange. And you can see that there's some overlap between these distributions. I mean, this is calcium imaging, which has some smoothing to it, and some of these frames are right next to each other, but you can kind of separate them. So to measure this separation, we'll convert these distributions to cumulative distribution functions, CDFs, and then we'll just plot them against each other. So we'll take the value of this CDF for attack versus sniffing, and that'll be our first data point down here, and then we'll go over from -5 up to 20 on this axis, and each point on this line is the value of the CDF of attack frames at a given delta F over F and a value of the CDF of sniff frames. So if these two histograms were perfectly overlapping, you'd get a line just along the diagonal here. But in this case, you get an increase in the number of sniff frames before you start to see attack frames, because you have all of these guys down here, levels of activation that are low that correspond only to sniffing and not attack. So any deviation of this area under this curve from 1/2 from this line y equals x means that you have some ability to distinguish these two behaviors, attack and investigation, based on just how active this cell is. So the AUROC, or the choice probability, is just the area under this curve integrating over all the values. And like the event-triggered averages, you can compute this for a whole population of neurons and create a histogram of choice probabilities and find cells where their AUROC, their choice probability, is higher than you would expect just by random chance. And I should say that chance here is just found by bootstrapping. You basically shuffle the timing of the attack and the sniff bouts, and you say, if I was just assigning frames to one of these two categories at random, how much overlap of the histograms would I get? You repeat that 1,000 times, and you look to see if you are higher than, say, 97.5% of them. And if you are, then you call that a significant attack-preferring cell. And if you're down here, your area under the curve is lower, that makes you a sniff-preferring cell. So just to give you a better intuition for how this behaves, the choice probability, the area under the receiver operating characteristic curve, will change based on the distance between the means of two distributions. So as the means move closer together, this area under the curve will decrease. And if you flip the means so that one is higher than the other, it will become less than 1/2. It also represents the variances of the distributions. So if you have the same difference between the means, but the distributions just become wider, so you have more overlap, then this also decreases. So it's a measure of not just how far apart the attack response and the sniff response are, but how much variance there is to those two responses. So it really is like, how reliably can you distinguish what behavior the animal's performing just based on this single neuron? So here's a couple examples of cells that had a significant preference for different behaviors in a hypothalamic imaging data set. We had a cell that was preferentially active during attack, another cell that was more active during sniffing and less active during attack, a cell that was a little more active during mounting and less active during sniffing, and a cell that was more active during sniffing and less active during mounting. So These cells, one by one, aren't going to perfectly distinguish these different behaviors, but you can at least see that there's some bias in which the cell is a little more active in one condition than another. The other interesting thing you can do with this kind of analysis is you can compare different brain areas. So if we have a histogram of choice probabilities of neurons in VMHVL, this aggression-related area, we can compute a similar histogram for other brain areas, and we can see that sometimes you have things that look very similar to each other. Other areas might have a very strong preference for a particular behavior where you can very clearly distinguish signals from each other. So it's a helpful way to kind of identify differences between brain regions, or differences between behaviors. If you have like mount versus sniffing and attack versus sniffing, you could see if there's a greater separation of one pair of behaviors versus another. You can also set a threshold for what counts as a neuron that is specifically activated during a behavior and create sort of histograms and look at what combinations of behaviors are represented by different cells and use this as another way to contrast different brain areas. In this case, we've kind of broken it down to cells that are preferentially activated during sniffing, sniffing females, males, interacting with females and interacting with males. And even though cell by cell or region by region, it might not tell you a lot, it's a useful tool for comparing different populations or different experimental conditions. Like you could do this in a hungry animal versus a sated animal and see to what extent the tuning of cells changes, or you could look in an animal that is wild type or mutant or normal and drug treated and see kind of how these response properties shift. Another way that's maybe a little more helpful for visualizing this is to just cluster cells by their behavioral responses. So what I've shown you, what I'm doing here is I took annotations for 14 different behaviors that had been manually scored for a series of imaging experiments. So we have annotations for when nothing is really happening, for consummatory behaviors, mounting and intermission or attack, and for investigation behaviors towards females and males. And we can cluster cells by their response to different combinations of behaviors. So for, say, medial preoptic area, a region that is heavily involved in control of reproductive behaviors, we can see that there are a bunch of cells that respond specifically to intermission and mounting, some cells that respond only to intermission, and also some cells that are specifically activated during other individual behaviors. We can also identify cells in this way that are activated just under broad conditions during multiple behaviors, like cells that are active anytime a female is present or anytime a male is present. And again, this is also really helpful for comparing different experimental conditions or different brain areas. So just as an example, if we do the same analysis in VMHVL, which is involved in control of aggressive behaviors, we see very few behavior-specific cells. We see a lot of cells that are just active in the presence of a male or in the presence of a female, regardless of the behavior the animal's performing. And you see that by just the coloration of these responses of the cells. I went through that pretty fast. Are there any questions? Is it clear what I'm showing here? I know it's like post-lunch and everyone's tired. I'll also add that to make these rasters, I z-scored the responses of the cells. So I took each column here is one neuron, and I'm showing the average activity of the neuron during each of these behaviors. And I z-scored them, which means I subtracted the mean and I divided by the standard deviation of the responses. So within a group of cells that's responsive to a behavior, some cells are going to be very strongly activated and others will be less strongly activated. But the relative difference in activation between behaviors is kind of what we're grouping these cells by. So that is looking at some single neuron properties. We can also use decoders and classifiers to see what kind of information can be read out by a neural population. So you've seen this diagram a lot now. We're going to be doing this again from the decoding side, which means that we have a stimulus feature or a behavior and we have recorded neural activity. And now we're going to look not just at what one cell is doing, but at what all of the cells are doing, and we're going to ask how well we can read out information about the stimulus or information about the animal's behavior. So you can do this with Fisher's linear discriminant analysis, which is what I showed you guys before lunch. There are a handful of other classifiers that you can use. Support vector machines are really nice. Naive Bayes, they perform fairly similarly if you're using linear methods. And in all cases, you're just trying to find a weighted combination of neurons that best reconstructs the signal that you're trying to decode. So a couple different strategies that you can use with decoders. The first thing you could do, okay, so you can take your observed delta F over F, your observed activation of a population of cells, and the behavior that the animal is performing, train a classifier to predict what behavior is happening given what the neurons are doing. The reason for a linear classifier we kind of discussed before lunch is that it's kind of like what a neuron is doing. It's taking a weighted sum of cells and thresholding it. But you can do other things with decoders to look at how information in a neural population is evolving over time. But first, right, when you are evaluating a decoder, we touched on this a little bit yesterday, you need to test it on a different set of data than you use to train it. So when you fit these weights, you're going to be picking them to best predict whether a behavior is happening or not. To evaluate it, you then want to take a held out trial or a held out chunk of time and have that decoder predict on that held out chunk of time. And for time series data, you always want to make sure that your training and test set are distinct from each other. So if you're not dealing with time series data, if each time point is independent, people will often just randomly assign data points to the training set and the test set. In time series data, these neighboring time points are highly correlated with each other. So if you randomly assign data points to training and test, and say you did like a 90-10 split, 98% of the points in your test set are going to have an immediate neighbor that's in the training set. And so basically it's not a real independent observation of performance. So just a note when you're training decoders to always evaluate them ideally on a different trial or a different animal, if that's something that's feasible, but at least a different chunk of time. Okay, so decoding current behavior from activity. You can do, I think you guys did this during the day one boot camp, predicting attack based on what neurons are doing, projecting neural activity onto a dimension and thresholding it. But you can also use decoders to ask when information is appearing in a system. So if you have, say, an imaged mouse and you're presenting a male or a female mouse to that animal, you can ask how long it takes for the imaged animal to determine the sex of the conspecific. So what you would do is take the time that the intruder mouse was introduced and look at the neural activity within a window of time. And for each separate time point, you can train a different decoder and say, given the neural activity 3 seconds before the animal was introduced, can I tell if it's a male or a female? And usually, ideally, you get something that looks like this. So before the intruder mouse is present, you can't tell if it's going to be a male or a female because your mouse isn't psychic and its neurons haven't received any sensory cues. But then once the intruder mouse is introduced, you can see that the activity very quickly achieves a separation of representations. You can distinguish based on how neurons are firing whether that animal is sniffing a male or sniffing a female. And you can show that that is consistently present in the population over time. So for these data sets, you need to treat each time point as a separate decoding task and only train based on that time point. This is kind of cool because you can look for appearance and disappearance of signals in a system. So if you have an attack bout that's taking place, you can try to predict sometime leading up to that attack bout whether the animal is going to be fighting or not. So this is kind of looking for preparatory activity that is an indication that the decision has been made to attack before the attack is actually annotated by the human who's scoring the experiment. And you can kind of go earlier and earlier in time and see if there's a signal that is predictive of this attack bout taking place. So in this case, you would take attack frames and non-attack frames, and see if your decoder can predict whether a future frame, like a second in the future or 5 seconds in the future, is going to be an attack frame or not. The other way that you see this used is to look for memory traces of inputs. So this is just the opposite direction. You take the onset of a stimulus or an impulse or sensory cue, and you look at the neural activity at some delta t after that, and you ask, given this neural activity, can I reconstruct what the input to this network was some number of time steps ago? So this is really useful if you're looking at working memory tasks. You can train for different time points after a stimulus was presented, train a separate decoder and see if that decoder is able to reconstruct the identity of the stimulus based on the observed pattern of neural activity. This is something I did really early on in my PhD, looking at memory traces of recurrent networks. You hit a network as an input and the activity kind of evolves over time. And which neurons are active changes. But by just looking at a fixed point in time, you can still distinguish based on that like time evolving pattern of activation, which stimulus was presented.
I have a question.
Yeah.
Are the kind of signatures of stimuli that reliable that you would be able to infer that?
It really depends on the stimulus. Yeah, eventually they're not. Like if you have a mouse that fought and then you look at the cell activity 5 minutes later, you're not going to know if it fought or not. But there is some time scale over which the signature of the stimulus or the behavior should still be present. And that depends a lot on the structure of the neural population. Some neural populations have a lot of persistent activity and their cell firing rates change very slowly over time. And so you could decode out what stimulus was presented 10 seconds or 30 seconds later. Other regions, you have very time-locked activity and it doesn't really last long after the stimulus. So it's kind of a way of getting a time scale of memory in different populations of neurons.
And then for the first example, would you have to distinguish between preparatory activity or kind of trait level signatures?
Right.
Of attacking or not?
Trait level meaning?
For example, if there's an animal, if there are a group of animals that are more likely to attack regardless of preparatory activity, would you have to kind of account for that? So the way I've presented it here, this is all decoding within the same animal. Because if we have a different animal, we have different neurons. You can try to be clever about it and like find an aligned representation across animals. That's something that Xuan is working on, training brain-machine interfaces that could be trained on one animal and used by another. And I guess for something like MRI, if you have like brain scale patterns of activation, you could do that maybe across individuals in a more reasonable way. Yeah, for individual cells, it doesn't make a lot of sense, but yeah, no, definitely.
Are you going to say more about how you treat each time point as
a separate thing?
Challenge?
Yeah.
I guess I'm, at least from previous research, you're saying used to thinking of, okay, you've got a block of time that is and isn't doing the behavior, and that's how you train it.
I hadn't prepared detail here, but it's basically like, say you have 10 time points, you're really training 10 separate decoders. So you take time point 1, you have a vector of each cell's firing rate on that time point, and you have, say, 50 trials, so you have 50 of those vectors. You train a decoder on that set of 50 trials and test it on that set, and then you do a separate decoder for 2 and 3 and 4.
So you need, it's that trial structure that's
You need, well, it could be, like here it's not a trial, it's like a trigger of an attack bout. So you could look at like when a jellyfish starts swimming, and you could look 3 seconds before that if there's any signature in the neural activity that is predicting that that swim bout is about to happen. No, it's a little fiddly to set up. You have to do this aligning and make. Usually these decoders are pretty fast to train, so you just run it in a for loop and train and test each decoder. So long as you're doing something linear, this is pretty straightforward. It doesn't necessarily mean that the same cell could decode the stimulus over time. Like it could be, if we go back to this example here, it could be that the male responding cells here are different from the male responding cells here. And so if I was a neuron that was reading out the population activity, I wouldn't get a consistent male signal. If you wanted to do that, then you would train like a single decoder on all time points instead of doing this time point by time point thing. Okay. As for choice of decoders, I've looked at this a little bit on hypothalamic imaging data sets. I just tried a comparison of Fisher linear discriminant analysis versus support vector machines. And there's not a ton of difference. You maybe get a little bit better performance for one versus another, but the trend across animals is the same. Sorry, this is just like a big data set of animals from the Anderson lab where we were trying this task. And if you do very poorly with one linear decoder, you're probably going to do poorly with another. You can try to boost performance with a nonlinear decoder or with data pre-processing steps, but among linear methods, it doesn't seem like the choice of decoder matters a whole bunch. Yeah, so some things to keep in mind just as general tips. For evaluating chance performance, the thing that I like to do is, again, bootstrapping. So I have neural activity and I have behavior, and I'm trying to predict, say, attack from what the neurons are doing. To see how well I would do by chance, I would shuffle these bouts of behavior and have attack bouts happening just randomly throughout this trial. And I could train a classifier on that too. And if I'm doing it right, if I'm not screwing anything up, that classifier should be at 50% accuracy. If it's not, that means that there's class imbalance or there's something else going on. And I need to go back and check the code or or report F1 score, yeah, or just be aware of that class imbalance and how it affects decoder performance. So rather than just, if it's a binary decoding task, rather than reporting things relative to chance as 50% accuracy, making a control decoder trained on shuffled data is a good way to get a sense of whether your decoder performance is significantly better than you would expect just randomly guessing. F1 score, I went through this really fast. F1 score is another way of computing performance of a decoder when your classes aren't balanced. Did you guys go over that guy? Adi says yes. So F1 score is, rather than did you get it right or not, say you have a behavior that happens 5% of the time, you could be 95% accuracy just by predicting that it never happens, but that's not a very great decoder. Instead, you report F1 score, which is a harmonic mean of precision and recall. Precision is true positives over true plus false positives. Recall is true positives over true positives, false negatives. It's basically, precision is if I have all of my true attack bouts, how many of them did I hit? Recall is I labeled these many things as attack, how many of them were actually attack? So if you label everything as attack, your recall is going to be low, but your precision will be high. If you label nothing as attack, your precision is going to be low and your recall is going to be high, I think. Yeah, so F1 score, sorry, this is rushing, but F1 score is another way of reporting accuracy that's worth looking into. So after decoding, there is measures of similarity. So rather than saying, can I distinguish these two things or not, you might want to say, how similar is the neural response to stimulus A versus the neural response to stimulus B? So in this case, we had a data set where we were imaging mouse responses to males versus females. And we saw that there were cells that were preferentially active in the presence of males and cells active in the presence of females, but we wanted to put a number on that and say how similar these states were to each other. So you can compute the average response of neurons to each of your stimuli, say a male versus a female, across periods of interaction or across a specific behavior. And then you want to come up with some way of measuring similarity of these things. And there are a couple ways to do this. Man, my slides are kind of out of order. The first that I want to talk about is Mahalanobis distance. So this is taking all of these male trials and all of these female trials and just asking how dissimilar they are from each other. So say you had a set of trials. So each blue point here is one stimulus presentation, and you have the response of a pair of neurons to those stimuli. And there's some correlation in how your neurons are responding. If you compute the Euclidean distance between a test point and the mean of this distribution, it's going to be equally high for a point that's up here versus a point that's out here, which is kind of not intuitive. You would think that if I presented a mouse and I got a response that was here, I'd say, yeah, it's probably pretty similar to all of these other stimuli that I presented, and it's probably another male or another female. Versus if I presented a mouse and it was evoked a pattern of activity out here, I would say, oh, that actually looks very different. I want to say that that's a very different response from, say, this point or this point. Sorry, it's Euclidean distance. In Mahalanobis distance, you measure your distance from the center of a distribution in units of standard deviation of the covariance matrix of the data. So if these are all trials where I've presented a male mouse, I want to treat this trial as being as close to the center as a point that's out here. I want to capture the fact that there's correlation between the firing rates of these two neurons, and recognize that moving along this dimension isn't as meaningful as moving along this dimension in terms of the identity of the stimulus. This is a method that's also commonly used for outlier detection. If I have a point that appears way out here, it's probably still part of this distribution, but if I have a point that lies out here, then it looks more like an outlier. Even though the Euclidean distance from the center is the same, it's very far from the rest of the points in this cloud. So in the Mahalanobis distance, rather than taking just the distance of each point from the center of the distribution, you take that distance and you normalize it by the inverse of the covariance matrix to scale by how much variance there is in different dimensions of the neural activity. So going back to our male versus female example, what you could do is take all of the trials where a male is present and all of the trials where a female is present, and then take a held out mystery mouse and ask how far is it from the female distribution versus how far is it from the male distribution in units of Mahalanobis distance. And what we find is that this gives us a very nice readout of how much a neural response resembles the average response to a conspecific. Another method that you'll see for measuring similarity between stimuli is the Pearson's correlation. So this is just taking the response to one stimulus minus the mean response to another, and like measuring the angle between these two vectors. So you have a point here and a point here. You measure how correlated they are. That's just sort of how small this angle between these pair of points is. This is something that sometimes makes sense because you want to say, is the pattern of activation of neurons to one mouse and another very similar, or are they kind of orthogonal to each other? But it behaves differently from a Mahalanobis distance. For example, it doesn't take into account the spread of points, the spread of responses to females or responses to males. So if we look at the Mahalanobis distance between males and females for different imaged animals, we see that it behaves a lot like a classifier for a certain range. So as you are better able to distinguish males and females from each other, the Mahalanobis distance between males and females grows. But then once you can perfectly distinguish males and females, your distributions of responses are very separate. You can further increase the Mahalanobis distance just by moving them further and further apart from each other, even when you're hitting perfect decoder accuracy. By comparison, if you do the same thing with Pearson's correlation, you often don't see a very nice correspondence between how correlated two patterns of neural activity are and how well you can distinguish responses to stimulus A versus responses to stimulus B. So we have some points out here where the Pearson's correlation is very small and the stimuli are easy to distinguish from each other. But in cases where signals are more correlated, it's Pearson's correlation between like the mean male evoked response and the mean female evoked response isn't really a good predictor for how well you could distinguish whether a mouse is male or female on a given trial, because Pearson's correlation isn't taking in any information about the variance of distributions. It's just kind of measuring an angle between them. That was kind of rushed. Are there any questions about that? You have a measure of distance in units of standard deviation versus Pearson's correlation is an angle between vectors. So I'm rushing a little bit, but before we go to the rest of the slides, I wanted to quickly do a MATLAB demo to talk about how our measures of distance depend on the dimensionality of our data sets. And I will post this code to the GitHub once I've translated it into Python so it's actually usable by people who code in reasonable programming languages. But I will share it for now. So what I've done here is I've made just Gaussian random, well, no, I need to. What I'm doing here is I'm creating just random signals, drawing from a Gaussian distribution. So I'll have 10,000 points that are sampled from a uniform Gaussian. And those points will be in 1-dimensional space, or in 2-dimensional space, or in 10-dimensional space. So think of it as like having the firing rate of a single cell that's Gaussian distributed versus 2 cells, each of which is Gaussian distributed and independent, versus 10 cells, each of which is Gaussian distributed and independent, versus 100 cells. And what I want to bring across here is how these measures of distance behave as the number of cells in your population increases. So here what I'm doing is I'm taking the response of, in blue is 2 cells, 10 cells, 50 cells, and 100 cells. And I'm just measuring the distance of points in this Gaussian cloud from the center of the cloud. So as you increase the dimensionality of your data, each point on average becomes further from the center of the cloud. But if you normalize by the mean of this distribution, you see that the normalized distance of points from the center collapses down to 1. So as your data becomes higher and higher dimensional, a Gaussian becomes effectively like a high-dimensional version of a sphere, where every point is about the same distance from the center as every other point. Whereas if you do this in 2 dimensions, some points are very close to the center and some points are very far from the center. So this is a very unintuitive thing that when you go into higher and higher dimensions, random noise, random distributions can behave very differently. I think the best way to think of this is if you're generating a Gaussian random variable 100 independent times, one of those times are going to be pretty far out in like one of your 100 dimensions. And it'll be different for different points. But on average, you're going to have a certain fraction of points that are far out. And that as you have more and more of these Gaussian random variables, you converge on a uniform distance of points from the center of a cloud. If we do this for different measures of distance, we get different results. So let's see if we have Pearson's. Nope. Pearson? No. I should have checked which ones were available. Let's go back to Euclidean. So Mahalanobis distance behaves pretty similarly to Euclidean distance. As your number of independent variables grows bigger and bigger, things tend to converge on being a uniform distance from each other, or from the center of the distribution. Let's check very quickly what other things, hang on. I thought we had a Pearson's, but it might not be called what I think it's called. Correlation. We can do this with Pearson's correlation as well. Ooh, that's interesting. Kind of, I think 2 is broken, but for the rest of them, the distance of points to the center collapses in to a kind of uniform level of correlation as things grow. An interesting point of comparison is a cosine distance, which is the cosine of the angle between vectors. This can also kind of collapse in an interesting way. So when you're creating measures of distance, it's worth thinking about what randomness looks like based on these different measures as your population of neurons grows or as your sample size grows. So measures of distance are very helpful for quantifying how similar things are to each other beyond what you can pick up using a neural decoder, but they're also a little bit tricky when you're dealing with high-dimensional data sets. Especially, yeah, I think if you look at things like Mahalanobis distance, it's very sensitive to how many neurons are present in your recording data set. And you want to take that into account when you're making comparisons, say, between animals or between imaging days where you have different numbers of samples. We'll go quickly through a use of PCA in making sense of neural data. PCA is kind of the first thing you try if you have a recording of neural activity and you want to see what interesting structure is there. And so say you do PCA, you plot your first two PCs against each other and you get some kind of spaghetti. It can be difficult to interpret what's going on sometimes, but there's a lot that you can do with principal component analysis beyond just plotting two PCs against each other, which is what you often see in papers. So I wanted to highlight an approach that was used in this C. elegans imaging paper from a couple of years ago. So they had an immobilized C. elegans in a microfluidic device, and they were presenting stimuli to the worm and imaging a bunch of neurons in the head, I think around 109 or so, and used PCA to make sense of what these cells were doing. And the process was a little bit more complex than that. They had this step where they took the derivative of the calcium fluorescence signals before they performed PCA. This is kind of a way of removing low frequency signals from your estimate of covariance between neurons, and is helpful sometimes when you're dealing with calcium imaging data where you kind of have this smoothing out of neural activity by your GCamp indicator. So they performed PCA on their neural activity. They projected the neural activity back onto those dimensions and took a look at them. They found that the first three principal components of the data set could explain a little over 60% of the variance, and that beyond PC number 3, each individual component explained very little. So they just took the first three principal components and they looked at what those signals looked like. And what they did that I think was really interesting was they used the loadings of neurons onto PCs as a way of organizing the neurons. So they got a matrix of neural activity that looks like this. So this is 109 neurons organized over time. And what they've done is when you're performing PCA, you have a set of weights by which your neurons are mapped onto your principal components. So PC1 is a weighted sum of all of your neurons, PC2 is a weighted sum, PC3. And they grouped cells based on, for each neuron, which weight was the highest. So these cells up here are the neurons that had the highest positive weight towards PC1 compared to that neuron's weight by which it contributed to PC2 and PC3. These cells here had the most negative weight contributing to PC1 compared to their weights towards PC2 and 3. This next group of cells had a high contribution to PC2. There's a small number with a negative weight on PC2, and then there were some PC3 cells down at the bottom. So once you do this sorting of cells by their projection onto principal components, or their contribution to PCs, you can identify groups of cells that have similar response properties. So it's kind of like k-means clustering or another method, but you're just looking at the signals that are kind of dominant in the neural activity, and you're using that to organize your cell responses. So this pulled out kind of 6 populations of cells, really mainly these 3, that had very different response properties and was a helpful way of grouping cells together for downstream analysis. And if you look at this, you can see different kind of patterns of activation that were present in the population that I think they found were correlated with movement of the animal. So there were these two populations that were kind of anti-correlated with each other and active at one stage versus another. And then there was the second population that really showed a distinct pattern of activation that was still a little anti-correlated with these guys, but independent and orthogonal to it. You can, as always, also project the neural activity of the population onto each principal axis and plot that as a function of time, which gives you another readout of what the population is doing that's a little easier to work with. So this brings up one important thing to take into account when you're working with PCA. I'm almost done. Which is that it allows neurons to have both positive and negative weights and make positive or negative contributions to principal components. So in this case, we really see that very strongly. We have neurons that contribute positively to the first PC and negatively. And these cells are anti-correlated with each other. So we've kind of taken a principal axis that looks like this, that's capturing the difference between how these two groups of cells are firing. In this case, it's kind of helpful because they seem so perfectly anti-correlated with each other. But if your cell activation is sparse, you can have pairs of neurons that aren't really doing anything in concert. It's just they never happen to be active at the same time. And you can find that PCA will apply a positive weight to one and a negative weight to another and kind of mix two independent signals into the same principal component. So PCA isn't guaranteed to really give you independent sources of variance in your neural data. It's just trying to find these dimensions that preserve as much variance as possible. Yeah?
Do you have a cutoff if like one of the components doesn't seem to be explaining much?
Yeah, so there's a couple ways you can do that. One is just to look at variance explained and look for this elbow where the adding in additional components isn't capturing very much. Another thing you can do is, well, one thing people do is they'll say, put a threshold on variance explained and keep that as enough components to get to that point. That's a little iffy because you don't really know if you need all of that variance or if some of it is just random spiking noise.
Well, especially there because it varies, like the incremental.
Yeah, it's very
small,
tiny.
But then, it does get to over 80, which I guess is
Yeah.
tempting.
Yeah. Another thing you can do is you can shuffle your data. So if you take this raster of neural activity, you can shuffle each neuron independently and then do PCA on that. And your variance explained will kind of look like that. It kind of falls off exponentially. And you only keep the components where their variance captured is significantly higher than you would expect in the shuffled one. The last thing you can do is project your activity onto each principal axis and only keep the ones where the projection is non-Gaussian, because Gaussian is kind of an indication that it's just kind of capturing random noise. And if there's structure in the projection, then that shows up as some non-uniformity in the projection. So everybody kind of has their own way of doing that. Usually you want to keep it also to a smaller number if possible, just for interpretability. And then another trick that they used that I wanted to highlight is this indication of direction of movement. So you can plot two principal components against each other like this, and time is kind of implicit here. Neighboring points in PC space that are connected are from neighboring time points in your data set, but you don't really know how the system is evolving over time. So the trick that they used in this paper was was to color the line by the direction of flow of the neural activity. So if you see a point that's green, it means that it's flowing in the green direction that you're seeing here. Pink means it's flowing in this direction. Purple means it's flowing in this direction. So you can use tricks like that to try to reflect time or direction of movement in PCA. You can see, the other way is just show a movie. You can see that the system really is taking these stereotyped trajectories where every time it's in a particular part of PC space, it's moving in the same direction, which is equivalent to seeing that all of these points are about the same color. And then the final trick I wanted to highlight was going back and taking individual neurons and looking at where their activity occurs in principal component space. This sometimes works and it sometimes doesn't. Just kind of have to try these things and see. So here's, again, the first two principal components plotted against each other. And these colored dots here are showing when a particular neuron is showing an increase in its activation. And what you see is that there are some neurons that are active in this lobe, some that are active in this lobe, and some that are active here. This is just 3 example cells. You can also zoom in on additional cells, and you can kind of see different clusters of neurons that are increasing or decreasing their activation at different points in PC space. So when you have your PCs, often you see some blob and you're like, yeah, that's a blob, cool. And you don't really know where to go from there. And by relating them back to what the individual cells are doing, you can sometimes start to find interesting neurons in your data set that you wouldn't have picked up on otherwise, and kind of start to come up with a story beyond just here's the principal components of my data. So that's pretty much it. I wanted to also point to a couple methods to use if you're still not happy with what unsupervised dimensionality reduction is doing for you. There are some semi-supervised methods where you're finding sources of variance that are correlated with other signals in your data set, like partial least squares. There's a method called demixed PCA, which is trying to find principal components that are correlated with different parameters of a task. Difference of covariances is a method you can use if you have 2 conditions and you want to find the components of neural activity that are most correlated with one or the other. And then, as we talked about before lunch, there are also methods for imposing certain constraints on your principal components. You can say you want them to be smooth in GPFA, or you can try to find components that have rotational dynamics, which is a method called JPCA. So that was a lot of things. Just to wrap up, and you can reference these slides later. There are lots of things you can do to try to make sense of neural data. And when you have a new data set, you kind of just go through a lot of these and try to see which ones stick and which ones don't. But each one kind of gives you a different perspective on what's happening at the level of single cells, or at the level of a population, or at the level of information that's present in the population. And by putting all of these things together, you can sometimes come up with a narrative of what your population is doing. So with that, are there any questions? Everyone's tired. I think I'll hand it off to Xuan and some practical work. Yeah. All right. Thank you, guys.